Most people think of antibiotics as a targeted intervention. You have a bacterial infection, you take the antibiotic, the infection clears, you move on. The reality is considerably more complicated. Antibiotics are not targeted, they are broad spectrum, designed to kill bacteria, and effective at doing so across a wide range of bacterial species simultaneously. When you take a course of antibiotics for a respiratory infection or a urinary tract infection or a skin infection, the antibiotic does not distinguish between the bacteria causing the problem and the bacteria that live in your gut, perform essential metabolic functions, produce neurotransmitters, train your immune system, and communicate directly with your brain. All of them are exposed. Many of them die. The gut microbiome, the community of approximately 38 trillion microorganisms that inhabit the human gastrointestinal tract, is one of the most complex ecosystems on Earth. It has been shaped over hundreds of thousands of years of coevolution with the human body. It performs functions that human physiology cannot perform without it. And a single standard course of antibiotics can disrupt it in ways that take 6 months to 2 years to fully resolve, if they resolve completely at all. Understanding what the microbiome actually does and what happens when antibiotics disrupt it changes how you think about antibiotic use, about recovery after a course of antibiotics, and about the connection between gut health and the brain symptoms, mood changes, anxiety, cognitive fog that many people experience after antibiotic treatment and almost never connect to the medication. The gut-brain connection is not a metaphor. It is a physical system with documented anatomical pathways, and understanding its basic architecture is essential for understanding why disrupting the microbiome produces effects in the brain. The vagus nerve, the same nerve discussed in the context of breathing and cold exposure, connects the brainstem directly to the enteric nervous system of the gut. Approximately 80% of the fibers in the vagus nerve carry signals from the gut to the brain, not from the brain to the gut. The gut is talking to the brain far more than the brain is talking to the gut. What the gut sends up this pathway includes information about the gut's chemical environment, the presence and concentrations of neurotransmitters, metabolites, and signaling molecules produced by the bacteria living there. The brain integrates this information continuously and adjusts mood, cognition, stress responses, and behavior in response to it. This is not a secondary or peripheral influence. Research over the past decade has established that the gut microbiome is a primary source of several neuroactive compounds, molecules that directly affect neural function, and that its composition strongly predicts psychological and cognitive outcomes in ways that were not anticipated when the first studies on the gut-brain axis were conducted. The most significant of these compounds in terms of brain function is serotonin. Approximately 90% of the body's serotonin is produced in the gastrointestinal tract, primarily by enterochromaffin cells in the gut lining. The production of serotonin by these cells is directly regulated by the microbiome. Specific bacterial species produce short-chain fatty acids and other metabolites that stimulate enterochromaffin cell activity and drive serotonin synthesis. When the microbiome is disrupted, when the species that produce these stimulating metabolites are killed or their populations reduced, serotonin production in the gut drops. The serotonin produced in the gut does not cross the blood-brain barrier in significant quantities, so it is not directly available to the brain as a neurotransmitter. But gut-derived serotonin plays important roles in gut motility, in the signaling that travels up the vagus nerve, and in the regulation of immune function in the gut, all of which have downstream effects on brain function and mood. The relationship between gut serotonin and brain serotonin is indirect but real, and it is one of several mechanisms through which microbiome disruption translates into mood changes. The antibiotic effects on the microbiome have been studied in detail, and the findings are more significant than most patients are ever told. A study published in Nature by Dethlefsen and Relman tracked the gut microbiome of healthy adults before, during, and after a standard 5-day course of ciprofloxacin, a commonly prescribed broad-spectrum antibiotic. During the course of treatment, the diversity of the microbiome dropped dramatically. Multiple bacterial species that were abundantly present before treatment became undetectable. The overall community structure shifted significantly within days. After the course of antibiotics ended, the microbiome began to recover, but the recovery was not complete. At the 1-month follow-up, most participants still had measurably altered microbiome profiles compared to their pre-antibiotic baseline. Some participants had not recovered normal microbiome composition 6 months later. A subset of bacterial species that were present before antibiotic treatment did not return at all during the follow-up period. A separate study examining the effects of a standard 7-day course of clindamycin found that the microbiome diversity of participants had not fully recovered 2 years after the treatment ended. 2 years from a single week-long course of antibiotics. These are not outlier results. They reflect a pattern that has been replicated across multiple antibiotic classes and multiple study populations. Antibiotics disrupt the microbiome significantly, recovery is slow, and for some bacterial species, the disruption appears to be permanent given the timescales studied. For most conditions for which antibiotics are prescribed, this collateral disruption is an acceptable trade-off. Bacterial infections can be serious. Antibiotics save lives. The point is not that antibiotic use is unjustified. The point is that the disruption is real. It lasts far longer than the treatment course, and the downstream effects, including effects on brain function, are not trivial and are almost never discussed with patients. The connection between microbiome disruption and mood, anxiety, and cognitive function runs through several overlapping mechanisms. The first is the serotonin pathway already described. But serotonin is not the only neuroactive compound the microbiome produces or regulates. GABA, gamma-aminobutyric acid, is the primary inhibitory neurotransmitter in the central nervous system, responsible for reducing neuronal excitability and producing the calming, anxiolytic tone that counterbalances the excitatory activity of glutamate. Certain species of gut bacteria, particularly Lactobacillus and Bifidobacterium, produce GABA directly. Research has shown that germ-free animals raised without any gut microbiome, have significantly altered GABA receptor expression in the brain and show behavioral patterns consistent with increased anxiety compared to animals with normal microbiome. Restoring the microbiome normalizes GABA receptor expression and anxiety-related behavior. Short-chain fatty acids, produced by bacterial fermentation of dietary fiber, cross the blood-brain barrier and directly influence microglial activity. Microglia are the immune cells of the brain, responsible for maintaining neural tissue, clearing cellular debris, and regulating inflammation in the central nervous system. Microbiome-derived short-chain fatty acids, particularly butyrate, are essential for normal microglial function. Animals raised without a microbiome have dysfunctional microglia, immature in form and abnormal in function. Disrupting the microbiome through antibiotics reduces the production of these short-chain fatty acids and impairs microglial function, which contributes to increased neuroinflammation and altered cognitive performance. The HPA axis, the hypothalamic pituitary adrenal axis that governs the stress response and cortisol production, is regulated in part by the microbiome. Germ-free animals show exaggerated HPA axis responses to stress, higher cortisol spikes, slower recovery. Specific bacterial species appear to modulate HPA sensitivity, and their disruption by antibiotics can contribute to dysregulated stress responses that manifest as heightened anxiety and reduced stress resilience. These mechanisms are not theoretical. They have been demonstrated in animal models and, increasingly, in human studies. A large Danish cohort study, published in the Journal of Clinical Psychiatry, found that antibiotic use was associated with significantly increased risk of depression and anxiety, and that the risk was cumulative with the number of antibiotic courses. People who had taken more antibiotic courses over their lifetime had higher rates of mood disorders than those who had taken fewer, with a dose-response relationship that is difficult to explain through mechanisms other than the microbiome. The cognitive effects of microbiome disruption are less commonly discussed than the mood effects, but the research is accumulating. The connection between gut bacteria and cognition operates partly through the BDNF pathway, the same brain-derived neurotrophic factor that muscle-derived irisin and exercise upregulate. Short-chain fatty acids from bacterial fermentation increase BDNF production in the hippocampus. Disrupting the microbiome reduces short-chain fatty acid production, reduces hippocampal BDNF, and impairs the synaptic plasticity that learning and memory depend on. A 2019 study in Nature Neuroscience found that transferring the gut microbiome from aged mice to young, germ-free mice produced cognitive impairment in the young mice. Their performance on learning and memory tasks declined toward the level of the aged donors. The microbiome, through mechanisms including short-chain fatty acid production and hippocampal BDNF modulation, was directly driving cognitive performance. The clinical correlate in humans is the cognitive fog, the subjective sense of mental sluggishness, reduced verbal fluency, and impaired working memory that many people report following antibiotic courses. It is commonly attributed to the illness for which the antibiotics were prescribed, or to disrupted sleep, or to general malaise. The contribution of microbiome disruption to these cognitive symptoms is almost never considered. Recovery of the microbiome after antibiotic disruption is possible, but it is not passive. Waiting for recovery without actively supporting it typically results in the slow, incomplete recovery described in the research literature. Dietary fiber is the primary substrate for the gut bacteria responsible for producing short-chain fatty acids. In the absence of dietary fiber, even bacterial species that survive antibiotic treatment cannot produce the metabolites, butyrate, propionate, acetate, that support gut lining integrity, microglial function, BDNF production, and the other downstream effects that matter for brain health. A diet high in diverse plant foods, vegetables, legumes, fruits, whole grains, provides the substrate diversity that supports microbiome recovery after disruption. Fermented foods have been shown in research to accelerate microbiome recovery and increase diversity more effectively than probiotic supplements. A 2021 study at Stanford University directly compared a high-fiber diet to a high-fermented food diet for their effects on microbiome diversity and immune function. The fermented food group showed greater increases in microbiome diversity and greater reductions in inflammatory markers than the fiber group. Foods in this category include yogurt, kefir, kimchi, sauerkraut, and kombucha. Any food produced through bacterial fermentation that contains live cultures. Probiotic supplements have more limited and inconsistent evidence than fermented foods for microbiome recovery, but specific strains have shown benefit in specific contexts. Lactobacillus rhamnosus and Bifidobacterium longum have been shown in controlled trials to reduce anxiety and improve mood in people with disrupted microbiomes. These are not universally effective, and the evidence does not support most commercial probiotic products for most people in most circumstances. But in the specific context of post-antibiotic recovery, targeted probiotic use alongside dietary changes has more support than either intervention alone. Sleep quality significantly affects microbiome recovery. The microbiome has its own circadian rhythms. Different bacterial species are active at different times of day, and disrupted sleep disrupts these rhythms. Prioritizing sleep quality during and after a course of antibiotics supports the microbiome's recovery timeline. And there is a specific consideration around prebiotic foods, foods that feed existing gut bacteria rather than introducing new ones. Garlic, onions, leeks, asparagus, bananas, and oats contain prebiotic compounds, particularly inulin and fructooligosaccharides, that selectively feed Lactobacillus and Bifidobacterium species, the bacteria most consistently associated with positive mood and cognitive outcomes. Including these regularly in the diet during microbiome recovery selectively supports the bacterial populations most relevant to brain function. Here is the synthesis. Antibiotics are necessary. They are, in many situations, life-saving. The point of this discussion is not to discourage antibiotic use when it is genuinely indicated, but to fill in a significant gap in what most people understand about what antibiotics do beyond clearing the target infection. The microbiome is not a passive bystander in human health. It is an active participant, producing neurotransmitters, regulating the stress response, modulating immune function in the brain, and communicating with the central nervous system through one of the longest nerves in the body. When antibiotics disrupt it, the effects are not limited to the gut. They extend to mood, anxiety, stress resilience, and cognitive function through mechanisms that are now well documented and that operate over a timeline of months to years rather than days. Knowing this changes what you do after a course of antibiotics. Not anxiously or dramatically, but deliberately. More fermented foods, more dietary fiber diversity, more sleep. The awareness that the mild mood shift or the cognitive fog or the heightened anxiety that follows antibiotic treatment may have a biological explanation, and that the explanation points toward a recovery strategy, not a permanent condition. The gut is not a separate system from the brain. It never was. It is part of the same integrated biology, communicating continuously, influencing each other in ways that medicine is only beginning to map fully. If this changed how you think about what happens after you take antibiotics, hit the like button. Share it with someone who has been through a course of antibiotics and never felt quite right afterward.